sunlight for photosynthesis. Now let's look at an animal cell. Human cheek cells are easy to obtain because they rub off during eating and after contact with the tongue, so they are perfect for viewing animal cells under a microscope. A cotton bud is used to rub very gently against the inside of the cheek. Smear the cotton bud onto the center of the slide to transfer the cheek cells. It might not be possible to see anything on the slide because the cheek cells are too small to see with the naked eye. A drop of methylene blue is added to the spot where the cheek cells were transferred. Methylene blue will stain each cell to make them easier to see. A cover slip is gently lowered onto the epidermis using a mounted needle for support. Air bubbles are avoided as much as possible. Excess methylene blue is removed using filter paper. The slide is left for 1 minute to give time for the stain to work. Set up the microscope as before. Remember not to let the lens touch the slide. The focusing dial is turned so that the stage moves away from the objective lens and stops when the slide comes into focus. Animal cells are often smaller than plant cells, so it might only be possible to see some small blue blobs with the lowest power objective lens. A different objective lens is selected to show more detail. Using a higher magnification, each cell looks a little bit like a pale blue fried egg. It might be possible to see the nucleus in the middle. Methylene blue stains the nucleus dark blue and the cytoplasm light blue. What are the main visible differences between the cells? The visible differences are: the onion cells are all joined together, but the cheek cells are separate. The onion cells are larger than the cheek cells. The onion cells have a regular, roughly rectangular shape, whereas the cheek cells have an irregular shape. The onion cells have cell walls, but the cheek cells do not.